Hi, today we will talk about the red blood cells size variations known as poikilocytosis or anisocytosis. These are not random, they often provide valuable diagnostic clues about underlying disease. So, first I will talk about the macrocytes. Macrocyte cells, as you see in the pictures, are the larger than the normal red blood cells. These are common seen in hemolytic anemia or liver disease or sometimes in hypothyroidism and chemotherapy. Their presence indicate either the increased cell turnover or impaired DNA synthesis. Now we'll talk about the hypochromasia. The hypochromasia red blood cells shows the increased central pallor due to the reduced hemoglobin. And this is the classic finding in iron deficiency anemia, reflecting impaired hemoglobin production. So now we will talk about the next image, which is the elliptocytes. We'll talk about the elliptocytes. Elliptocytes are really elongated, pencil-shaped cells. They appear in hereditary or elliptocytosis. Some cases in iron deficiency anemia or hereditary elliptocytosis. Now we will talk about spherocytes. Spherocytes are small, dense and lack central pallor. They appear in autoimmune hemolytic anemia, post-transfusion as well as in hereditary spherocytosis after blood transfusion. Their reduced deformability make them prone to splenic destruction. Now we will talk about ovalocyte. Ovalocytes are oval-shaped red blood cells are often seen in myelodysplastic syndrome, thalassemia syndrome, and megaloblastic anemia. Their presence indicates defective erythropoiesis or abnormal red blood cells membrane stability. Now, next we talk about target cells. Target cells are podocyte cells, shows a central bull-eye appearance. They are found in hemoglobin C disease, a liver disease, post-splenectomy states, and thalassemia as well as in iron deficiency anemia. They indicate abnormal hemoglobin distribution, excess cell membrane relative. Now we will talk about the sickle cells. Sickle cells are drepanocytes or crescent-shaped red blood cells due to the abnormal hemoglobin S polymerization. They are diagnostic of sickle cell anemia and are responsible for hemolysis in the blood. Now we will talk about Burr cells. Burr cells are echinocytes so short, evenly spaced projections. They occur in renal disease, liver disease, and severe burn cases. Unlike acanthocytes, their projections are uniform and reversible. Now we will talk about the schistocytes. Schistocytes are the fragmented red cells. Look like platelets. They are marker of the microangiopathic hemolytic anemia, prosthetic heart disease, severe infection like Clostridia, and pulmonary embolism. Now we will talk about the helmet cells, particularly seen in G6PD deficiency, oxidative damage, and pulmonary embolism. They signal a mechanical as well as oxidative destruction of RBCs. So, in summary, variations in red blood cell size and shape are really very powerful diagnostic clues. Whenever you examine the slides, you always carefully examine the peripheral blood smears, helps in identifying underlying conditions such as nutritional deficiencies, some hemolytic disorders, membrane defects, systematic disease. Each cell morphology tell its own story in clinical pathology. So next, when you examine the slides, so always use the thin smear to check the RBC's size, shape, and all morphologies. Like and subscribe Pathoscope Lab.